Welcome everyone to the first Soils Network of Knowledge webinar for 2019, and welcome particularly to those people who have not attended one of our webinars before. So I'd like to introduce the New South Wales DPI Soils Unit's Mark Whatmuff. Mark specialises in investigating the impact of heavy metals and other contaminants on agricultural production, the soil and the wider environment. His research to date has centred on assessing the effects associated with a range of organic waste amendments applied to field soils. And this work has contributed to the formulation of regulatory guidelines designed to allow the safe reuse of organic amendments in New South Wales. So that's why he's going to talk to us today about the benefits and risks of using recycled organics, which is a very topical thing, as you can imagine.
Thanks, Abby, for that introduction.
No problem.
Good morning, everyone. Yeah, look, my name's Mark Whatmuff. I'm a soil chemist with New South Wales DPI. Quite a few of you may know who I am, and I've probably worked with quite a few of the people listening at the moment, but let me assure you that that photo is quite old. Okay, so I'm going to talk to you today for a while, looking at talking about some of the stuff, some of the work we've done at DPI on recycled organics over the past 20 years plus, with some reference to how that then stacks up against some of the information that's coming out of the literature on similar materials. So most of what I'll be talking about today is centering around those four recycled organic materials: green wastes, biosolids, manures, and mixed waste organic outputs. I'll be talking about some of the possible benefits that you can get from using this stuff on soil, and some of the risks that might occur if we're not careful. But now for something completely different. I guess one of the things that underpins a lot of the work that researchers such as those from DPI and elsewhere is that what we try and do our work in a manner that's statistically viable. So you'll see in various research papers statements like a complete randomized complete block design, or we used a repeated measured analyses, ANOVAs with data was transformed, least significant difference. Lots of what appears to be a lot of jargon, but what this actually means is that the trial work we do is statistically designed to allow us to compare treatments, and that any effects we see are not due to chance. So this means that we can compare treatments, we can compare interactions between treatments, how these change over time. It means that our work is reproducible, or hopefully it's reproducible, but it also gives credibility to some of the conclusions we draw from the data, and also including our ability to predict what may happen next. And I had a professor way back when I was an undergraduate who said to us that if your data is not publishable, it's hearsay. So that's one of the reasons why when people such as myself put up data and talks, we do have place a lot of emphasis on whether it's statistically significant. And the reason for that is that it allows us to actually present viable conclusions from the data. All right, so why would we use recycled organics? What are the benefits? So the Green Revolution was actually not that long ago. In the 1940s and 1960s, we started using inorganic fertilizers, but before this, organic fertilizers were what we used. But we found that we weren't able to feed the growing population, and therefore we needed inorganic fertilizers. But we've come full circle to some degree, because at the moment what we want to try and do with recycled organics is to reverse the net drain of plant nutrients from our farmlands to the cities. And we know that a lot of the recycled organic materials that we have access to have a range of nutrients, the most important being nitrogen and phosphorus, but there's also some sulfur and potassium and calcium. We're also looking to recycle and improve the carbon status of our soils, and a lot of these materials, recycled organics, has carbon in it. There's also some issues where some of them may have trace elements, which may be of benefit. And we're trying to improve soil health with the overall package of using ROs on our soils. So when we started looking through some of our results and some of the work that we've done, when we compared it to what has happened in the literature, we found that, you know, some of the stuff that we've been finding on our soils are actually been found before in other parts of the world. And so I've just listed some of the sort of light bulb moments we've come across with some of the RO work we've done. And one of the early things we found is that the use of recycled organics doesn't necessarily always raise yields, given that there's enough nutrients there already from inorganic fertilizers. And there's been a recent very good meta-analysis of organic amendments conducted, published last year, where they found that continuous use of recycled organics didn't boost yield compared to the standard inorganic fertilizer. But when they both were together, things were better. And they found that inorganic fertilizer as a supplement to the RO aided the breakdown of the organic matter, and that therefore released the nutrients that those materials contained. The study also found that the types of waste were very important, application rates and matching them to the soils. We've generally found in the work we've done, and this is backed up by the literature, that high applications of RO are often needed to exceed standard practice. And for example, some of the work done at DPI found that we needed at least 100 tonnes of compost, sometimes plus urea, to exceed the standard practice. And one of the reasons that that some of the RO, there's variability in the response of the recycled organics, is the differences in nitrogen mineralisation. And obviously these materials are quite variable, and their availability is sometimes lacking. So by and large, this kind of this variability has generally slowed and retarded the take-up of these materials as a coverall in agriculture. In terms of nitrogen mineralisation, it's clear that one size doesn't fit all. We're still not that good at predicting nitrogen mineralisation from recycled organics, but our tests are getting better. Ultimately, the best test is to incubate the material in the field, because when you then combine climate, soil type and waste properties together, it's very difficult to do that kind of study in a lab, but we're certainly moving there. And I'll give you an example. So some of the biosolids work we did, we had a range of between 30 and 80% nitrogen mineralisation in the first year after the biosolids were put to ground. We've also got issues where sometimes there's a lag in the field response, particularly with yield, because it takes a while for the microbial population of that soil to catch up. And this is another area where perhaps supplementing recycled organics with inorganic fertilizer may aid this process. So this is a table of some of the recycled organics that I've looked at, and I've listed here the nutrient contents, phosphorus, carbon, nitrogen, and carbon-nitrogen ratio. So it's interesting to see that really some of the fresher, less composted materials, stuff like biosolids and fresh manure, has a higher carbon and higher nitrogen content compared to some of the more composted materials. So we've got materials like a range of different biosolids from across the country. Some of them are anaerobically digested, some of them are dried. We've got different kinds of cattle manure depending on what the particular industry is. We've got pig manure, a couple of different poultry manures, including quail. That was pretty exciting. We have mixed waste organic outputs. That's the MWU, some green waste, and some composted biosolids. And what we note firstly is that when we dry a material, for example, if you look at the difference between the dried and the standard beef cattle manure, we actually lose a lot of the nitrogen and phosphorus in them. But interestingly as well, the carbon-nitrogen ratio doesn't change. Same can be said for a biosolids. We lose a lot of the carbon and the nitrogen when that material is dried. For some of the more composted materials, we also see that the carbon-nitrogen ratio is much higher than if the material hadn't been processed other than digested. And we want to have a lower carbon-nitrogen ratio if we want to supply nutrients to the soil, but we want a higher carbon-nitrogen ratio if we want to supply carbon to the soil. So not all RO materials are the same. They have different carbon contents, different nitrogen contents, and we need to match those to the end use. And one of the things that can happen, I mentioned earlier, that we can sometimes have a lag in the time for the soil microbial population to catch up with the addition of these organic materials, and that can then be translated into a lag in yield response. This is some work we did on biosolids under the National Biosolids Program a number of years ago at our site here at Camden. The topsoil is the night paddock soil, which is the green line, is a fairly fertile soil that's been under pasture for a number of years, whereas the blue line is the flat paddock on our site here, which is very poor nutrition, very badly managed, and is really quite in a poor state of affairs. So what we're seeing here is the response of these soil microbial population to in its ability to process added nitrogen that we've added, and it's called a substrate-induced nitrification test. It's a standard microbial test where we look at the soil's ability to assimilate an added nitrogen substrate. So when we've added biosolids to these soils, one soil, the green soil from the night, or the green line of the night paddock soil, has responded fairly well and has used up most of the substrate that we've put on, whereas the poorer soil, when we put on the biosolids, has not coped, especially when the higher end of application has happened, has not been able to assimilate those materials we put on. So there's definitely a case for targeting soils and being expectant that the actual response to these RO materials may not be straight away. All right, so let's talk about some yield effects when we've used these RO materials in the past. So this is a trial that was conducted at our grower site here at Menangle, where we compared the yield response for a range of different organic materials. We have mixed waste organic outputs, some green waste, biosolids and manure, incorporated and surface applied. So this is the second crop after the material was put on the ground, and we're looking at wheat yield on the vertical axes. So on the left-hand side of the graph are the actual data points with the treatments, the right-hand side of the controls. And we found that, firstly, the yield response to surface applications was much lower comparatively compared to whether the material was incorporated. But by the second year after we put this material on the ground, we needed it approximately, or greater than, 60 tonnes a hectare of green waste or municipal mixed waste organic outputs to get an increase in yield compared to the control. And by the second year, none of the surface treatments was better than the control treatment. By year 3, so that's 3 years after the material was put on the ground, only the 60 tonne plus treatments for the mixed waste organic outputs exceeded the control, but all the manure treatments had come in. So we've passed that lag phase with a high nitrogen containing RO has been now accepted by the soil microbial population, and things are kicking on. But the important thing to note is that we needed to put a fair bit of this material on to get a yield response. All right, so we do like to get carbon from recycled organics, but recently there was a meta-analysis published by the University of Sydney where they found that a 1% increase in soil carbon only gave a very small increase in soil water holding, and that this was mostly occurred at the wet end of that range, and that's more pronounced on a sandy soil. And that's because the particle size of the carbon material that we're putting on is much bigger than the clay or the smaller soil particles, therefore it will hold less water. So using the carbon contents that we discussed earlier for those waste materials, ranging between 8 and 36%, assuming that half of this material will persist in the soil, and using the 1,000 tonnes of soil per hectare, we need approximately, well, between 55 and 260 tonnes of recycled organics to raise soil carbon by 1%, depending on the material. That is a lot higher than we would have expected. But when we actually did our own moisture holding capacity on those soils for the trial that I mentioned earlier, where we compared mixed waste organic outputs, green waste, biosolids and manure, we did find that we needed very high rates, once again, to get an increase in soil water holding above the control. And in actual fact, once again, it was at least 60 tonnes of mixed waste organic outputs and more than 200 tonnes for green waste. And that's once again because of the particle size of that material is not conducive to holding water. Now, incidentally too, there was no effect on soil water holding from manure or biosolids. And we think one of the reasons for that is that the carbon was quite transient for those materials, and I'll talk about that in a minute. So, talking about the carbon from recycled organics, we did manage to increase the carbon with our treatments, and it was particularly important for a higher impact for the surface treatments. Once again, this graph shows the change in total organic carbon in the soils on the vertical axis, where we've got application rates on the y-axis. We've got the different treatments, the circles representing the incorporation treatments, the triangles representing the surface treatments, comparing mixed waste organic outputs, green waste, biosolids, manure, compared to controls, the control levels on the left-hand side. And the first thing we noticed was that the total organic carbon content of the control was greater than the control for all treatments above 60 tonnes a hectare, but not for biosolids. Not for the composted biosolids anyway. And for the surface treatments, we only needed 10 tonnes a hectare to raise our total organic carbon levels above that of the control. But how long did this carbon last? So the figures you're looking at right now is for the initials immediately after we put those materials on the soil. After 3 years, 25% of the carbon in the green waste treatment had disappeared, and 55% of the carbon in the manure treatment had disappeared, and 45% of the carbon from the mixed waste organic outputs had disappeared. And we can see that graph there on the right-hand side showing that the loss of carbon for each of the MWU treatments at the various rates, cumulative loss over the 3-year period. But we also know that the loss of carbon was much greater for the manure treatment than, say, for the green waste, and the biosolids didn't really lose a lot of carbon as well. So both the MWU and the manure treatment lost a fair bit of carbon in that first 3 years, but it was much greater, that decomposition, when the carbon was placed on the surface compared to when it was incorporated. And this graph shows the change in carbon in percentage for the different application rates for MWU, and with the blue diamonds representing whether the material was incorporated, and the red whether it was surface applied. So almost double the carbon was lost from the surface treatments compared to when it was incorporated into the soil. All right, let's talk about some risks. So a while ago, there was a paper that came out in the Australian Journal of Soil Research titled "Is the Soil an Appropriate Dumping Ground for Our Waste?" And this was around about the time when a lot of waste materials was beginning to be diverted from landfill and was heading towards being used and recycled on the land. But the conclusion that they drew was that we need to manage these recycled organic inputs carefully to minimise these risks. So some of the risks principally arise because these different materials come from different waste streams. There are a range of inorganic contaminants, mostly metals, but there's also a range of organic contaminants, including phthalates and pesticides and plasticizers. There's also the issue of physical contaminants, and these risks are different for different materials. So I can talk to you now about some of the different metal contaminants found in a range of ROs. And the first thing to note is that we've done a lot of work in the past on biosolids. Biosolids are very heavily regulated around the world, but a number of the metal levels in other waste materials are of the same order of magnitude as you would find in biosolids, particularly things like intensive manures, where they use zinc and copper as feed additives. We can see here that with the range, for example, of zinc in biosolids ranges between 180 and 1700, we're seeing some of the cattle manure and some of the pig manure up around 1000. So to suggest that manure is cleaner than biosolids is perhaps not quite right. There certainly are risks with all the different RO products that we want to use. And when we actually look at some of the composted materials that we've used in our most recent trial, we see the same thing occurs. So we've got a couple of the mixed waste organic outputs here compared to a green waste and a composted biosolids. We can see, for example, that the zinc is comparable or higher than those found in the biosolids for our MWU treatments. But also too, we find a range of organic contaminants in some of the mixed waste organic outputs that we won't find in other waste materials. So the take-home message is that we need to be aware of what's in the material before we put it on the ground, and to assume that because something comes from what we think is a clean source doesn't necessarily mean that it will not contain contaminants. So one of the things that we noticed very early on is that some of the materials we use are quite salty. In particular, manure and the mixed waste organic outputs were quite salty. This graph shows the change in soil EC, so electrical conductivity, for increasing rates of applications of the mixed waste organic outputs, the green waste, the biosolids and the manure, compared to the control soils on the right-hand side of the graph. We can see firstly that surface applications have a higher impact on EC per weight or per amount applied compared to whether the material is incorporated. But the manure treatment was quite salty, as was the MWU, and we breached the critical, some high salinity criteria at rates close to around about that 60 tonne a hectare. We were rates close to or just above where we would first start to see an agronomic response to this material. So let's talk about some metals. So given that we need to put a reasonable amount of this material on the ground for a number of the RO materials, it doesn't take a lot to start to approach some of the guideline or the soil contaminant limits. And we find, for example, with the MWU that we used, one agronomic application, we start to get close to the limit for cadmium in the soil. Whereas for some of the cleaner materials, we don't really see the same impact, for example, the green waste and the manure. But also to note as well on the right-hand side of that graph that when you apply this material on the surface and don't incorporate it, those contaminants are much more, have a much bigger impact on the soil concentration than would otherwise be the case. So once we need to be mindful too that when we put contaminants on the soil, a number of them persist. There's no evidence that the metals will disappear from the soil, and if they do, it means that they've leached and have gone somewhere else. But in particular, some of the organic chemicals, we are assuming that they will break down, but in the case of our data, we found that some of them have persisted for quite a significant amount of time. And I want to focus here particularly on the BPA, the bisphenol A, that we found in the soils following the application of the mixed waste organic outputs. BPA is a plasticizer. It's regarded as potentially harmful to human beings, but we are told that it has a pretty short half-life in the soil. And when we actually look at some of the soil data, and this graph shows the change in bisphenol A concentration in the 3 years following application of the MWU treatments to our soils. We have the time T0 at the top, a year later at T1, and 2 years later at T3, with application rates on the x-axis and the bisphenol A concentration on the y-axis, noting too that that's a log scale. There is some degradation, particularly if we look at some of the lower rates around about 50 tonnes a hectare. Once again, the surface treatments were higher comparatively compared to the incorporation treatments. But there's also some persistence, particularly at the higher end. That bisphenol A is still in the soil at concentrations not that much lower than when we first started, given that the half-life of bisphenol A is supposed to be between 4.5 and 7.5 days. We don't really know why that material hasn't broken down, but there are other chemicals that we could have listed that are doing exactly the same thing. So one of the things that happens when we put recycled organics on the ground is that we can reduce the volume of runoff. And this is some work that was done by Willie Joshua when he worked for DPI back in the early 1990s. And it shows the change in runoff yield for after different rainfall events at different rates of biosolids application. So we put more biosolids on the ground, we get better growth, better surface roughness, and less runoff. And the same thing was found when we did our trial here at Menangle with the mixed waste organic outputs. We put the more material we put on the ground, we did see a trend where runoff yield dropped. And this graph shows the change in runoff volume for the surface applications in the triangles and the circles representing the incorporation treatments compared to the control on the right-hand side. So we did see a general reduction in runoff volume with increasing rates. Unfortunately, that runoff from those particular treatments increased in concentration of the nutrients and contaminants that were leaching off those soils with increasing rates of application. This particular graph, fairly topical at the moment, looking at nitrogen runoff, but this is the data we got for the mixed waste organic output treatments for the surface applications in the triangles and the incorporation treatments in the circles for increasing rates of application between 0 and 200 tonnes compared to the control on the right-hand side. Once again, the concentration in the runoff from the surface treatments was considerably higher than when the material was incorporated. And even when we accounted for the differences in volume, we did find that the amount of material leaving the surface treatments was higher than those from the material that was incorporated. So there's also potential for some of the contaminants that we put on the soil to adversely affect soil biota. And certainly in Europe, soil biota and soil microbiology is a big issue, and it's certainly a very sensitive indicator of soil health. This particular test is worm avoidance, one that's favoured around the world as a very early indicator where ecosystem services might be threatened by contaminants or various issues in the soil. The premise is that worms will avoid conditions that they don't like. You have a chamber with a control soil on one side and a treated soil on the other. You put worms in the middle, and then they will move to the side that they prefer, and you then get a number where you can assess whether they've left some adverse conditions or otherwise. And we conducted those tests on a number of the materials that we've used at our site here at Menangle, and this particular one shows the worm avoidance behaviour seen with the mixed waste organic outputs when it was freshly applied to our soils. And we saw that we had worm avoidance at rates equal to or below those where we would expect to see an agronomic response to the material that was applied. And that's at around about 20 tonnes a hectare, we started seeing worm avoidance in these treated soils. Tests conducted on the same soils a couple of years later showed that the worm avoidance behaviour had dissipated, but in the meantime, if that was a real world situation, I don't know really where the worms would have gone. Nonetheless, fresh applications can interrupt some of the ecosystem biota, particularly in this case, things like earthworms. And we've also found that there have been some changes and some effects on soil microbiology. In this particular case, we're looking at the changes in soil respiration in terms of the amount of carbon dioxide evolved by the soil microbial populations, where we've actually tried to tease out different microbial populations that respond to different sources of sugar, food source. And we can profile some of the changes that may be happening when we look at the differences in responses to those added sugars. When we looked at the material that was freshly applied, most of the responses to the added sugars, the various added sugars, was positive. You would expect an impoverished soil that's received various recycled organic materials to respond positively because there are a number of very readily available food sources. But sometime later, some of those same soils showed a decline in respiration response in some cases that were below the control level for those same sugars. We do think that there is some changes, some issues going on with the microbial populations at this site with the application of some of the RO materials that we've used. All right. So I've very briefly talked now about physical contaminants. There are a number of physical contaminants that we can find in our recycled organic materials. Some of them have different contaminants than others. In this particular case, we are asked by the EPA to assess what would happen, what is the impact of applying crushed glass to soils, because we know that there is some glass in some of the contaminant materials that we use. So we're actually looking at the difference in under a microscope between some of the mill glass and the sand. Sand is a very different material than glass, and it's fairly obvious the glass is quite jagged. But nonetheless, there are some materials that contain crushed glass, and there are regulations that allow for a certain amount of glass to be applied to soils under various standards, and these materials are actually modified to make sure that the glass meets these standards. But when we grow plants on soils with this added glass, we can end up with glass on the surface of these plants when we take them out of the soil. And that's not necessarily something that we particularly want to see. We didn't assess the impact of ingestion of glass by grazing animals, but that's something that's certainly of concern to our animal specialists. All right, key findings. I think it's fairly obvious that recycled organics can improve crop yields, but it's also quite evident from our work and work overseas that often high rates are needed. Similar is the case for when we're using recycled organics to boost soil carbon. A lot of that carbon isn't going to be there for very long, and to actually get a meaningful change in something like water holding capacity, we once again need to add a lot of material there to change that. So can you actually get a carbon response and a nutrient response in the soil at the same time? Are they actually compatible? I think that that's quite difficult to achieve, and I think you need to match the needs of the various material you use to the endpoint. If you want to apply carbon, then you use something where that carbon isn't going to disappear. If you want to apply nutrients, you want to apply something with, say, a low CN ratio where you're going to get nutrients supplied to the soil. This whole process is very much soil type dependent and climate dependent, and I don't think we're across being able to predict exactly what these things are going to do all over the place. And I think one of the reasons for that is that there's often a lag in response to how the soil deals with this material when we put it on the soil, in particular the soil microbial population. And there's probably a case for trialling the use of inorganic fertilizers as a supplement to be applied with the RO. That will then enable the more rapid and steady breakdown of the organic material in the RO, thereby releasing the nutrients that it contains. So unfortunately, if we do have to add material at the high application rates, then there can be an issue with some soil contaminant levels, and these can have a potentially detrimental effect on soil biology. We've clearly shown that it's definitely better to incorporate the recycled organic material and don't leave it on the surface. You get a much better yield from your carbon and your nitrogen that's incorporated in the soil. You get less and slower breakdown, and you get less exposure to those contaminants. And we must be aware of what's in the RO material before we put it on the ground. Okay, that's enough for me. Thank you.
Thanks very much, Mark. There's a lot of information in that, and
oh, look, that's 25 years. Yes, yeah.
So, as I said, there is now time set aside to ask for any questions of Mark or points of clarification or comment. Just start with one that I had around if you'd like to make comment, Mark, on the difficulty or the likelihood of using recycled organics as an improver for poorer quality soils, given what you said about the rates and the risks involved with the rates required.
Yeah, look, that's a really good question. I mean, intuitively, you wouldn't expect to see, for example, a response from recycled organics on a beautiful vertisol at Narrabri, because it's got most of what it needs. It's probably ticking along quite nicely, thank you. And so intuitively, you would think the poorer soils would benefit from using recycled organics, but I view it as it's similar to feeding somebody a roast dinner after they've come out of a period of feed deprivation, and they can't necessarily assimilate those nutrients in that roast dinner, and it might even make them sick. We find a similar thing when we lime a soil. We will get a change in pH, but whether the soil microbial system will catch up with that in due course is another matter. I think too that poorer soils tend to be less capable of assimilating contaminants as well. Look, eventually they will change. That slide I showed earlier where the respiration on the poorer soil dropped off as we put the biosolids on. 2 years later, it was back to 100% use of that substrate. The soil did respond, but it's going to lag and be slower than if the soil's in reasonable shape.
Okay, thank you. Now we have lots of questions from people. I'm just going to go through these as they've come in. Michael would like you to explain the LSD stuff around the statistics, I'm assuming.
Okay, so LSD is the least significant difference. So it's a measure of the variation between treatments or means of treatments where they are not, or they are significant different from each other. So if the difference between treatment A and treatment B is smaller, the average of treatment A and treatment B is smaller than the least significant difference, then it's not significantly different. So it's a measure of whether there is actually a statistical difference between treatments.
Great, thank you. And if you're interested in anything around that, Michael, further, we actually did a webinar on statistics stuff around field trials with our biometrician here at DPI. I can't recall if that one's up on the web yet, but if you want access to that, just let me know and I can give you a link.
And look, can I also say in regards to that, so you'll see sometimes you'll see presentations where there appears to be a trend in the data, whether it's dropping, whether it's increasing, and you'll see sometimes there are error bars on the data points. So just because the line looks like it's moving up or down doesn't mean that that's actually no different than, not that difference is not due to chance. So if the error bars overlap, for example, the chances are that they're not different, or if that difference between point A and point B is smaller than the LSD, then they're actually, that difference is not due to a treatment effect, it's due to chance.
Yep.
So there's a difference between a trend and whether something's actually statistically different.
Okay. There's a few questions about subsoil applications. So there was one about subsoil manuring and whether that process would bypass some of the risks that you've talked about. And Andrew Dougal said subsoil amelioration can deliver higher yield responses with lower rates, around about 20 tonnes per hectare. He said, is that a viable option? So could you deal with those two questions together?
Yeah. Look. I think anything that gets these materials off the surface is a good thing, because you're minimising, for example, gaseous loss of nitrogen, You're minimising gaseous loss of carbon. You're getting more of a chance for that material to interact with the microbial population, which is not on the soil surface, in the soil. Remembering that the soil microbes are what actually cycles this stuff. You've got to then balance, though, at how deep you want to put it. And it's quite expensive, as you know, to put stuff into the subsoil. And I'm talking 30 centimetres or 20 centimetres. That's quite difficult to do. So that's quite a difficult option. We did have a program, or Sydney Water did have a program a long time ago where there was deep subsurface injection of biosolids. That was a liquid material, and that's when governments had a lot of money to spend on this stuff. And to be honest, that was brilliant because it was wet, it was under pressure, the material was, because it was under pressure, it really did affect a lot of the soil and impacted on it very well. Look, I think if you can afford to get it into the soil, it's a good thing. But it's whether it's actually cost effective, that physical process of getting it into the soil at 20 centimetres. And that's different from actually incorporating it, whether you're disking it in or using a harrow or something. But look, anything that stops it being left on the surface, you'll end up capturing more of the nutrients and carbon that you paid for when you got the stuff there in the first place.
Great, thank you. And now there's also a few questions about contaminants and how we could perhaps improve recycled organics to reduce contamination. So one of the questions was, what would you want to exclude, and then how can we improve that? Is that, do you feel like that's within your area to comment on?
Yeah, look, I don't really know what I would like to exclude means. Look, I think that you've got two schools of thought. You've got zero impact, zero effect, and that's just not possible. There is no landscape on Earth that we have not impacted in some way, shape or form. But you don't want to leave a legacy where a soil's land use capability is damaged or reduced just because you want to prevent a material ending up in landfill. And they're the two absolutely brutal ends of the scale. I think, for example, with biosolids, a lot of what the biosolids we get now are very, very clean in terms of contaminants compared to what they were 30 odd years ago when I started working with them, because you just can't get some of those contaminated materials anymore because trade waste, Sydney Water and so forth, have actually then gone to industry and said, look, you can't keep putting this into the material. I think there's definitely scope for that. There's definitely scope for more incentives for source separation. I think, for example, animal industries can look at feed additives and so forth. So I think we can actually start to reduce the impacts of these things going into our recycled organics. And I think there may well be some recycled organics that we really shouldn't be using on our soils.
Yep. Okay. Thank you. Okay. Now there's a couple of questions that relate to how nutrient and carbon levels change within products as they dry out. So Steve Kimball was interested in the difference in phosphorus levels and whether you had an idea on what the mechanism for that might mean as manures dry out. And then Mickey Wang said, why does the carbon and nitrogen in the compost drop so much when they're dried? So they're those two questions. Okay,
so they're really the same thing. Yep. Look, I really don't know what happens to the phosphorus. I'm not a phosphorus chemist. Look, I think a lot of the phosphorus that's in the recycled organics is an organic form. So I'm assuming that as the carbon volatilizes and breaks down and disappears, that some of the phosphorus will disappear in that. But look, I'm only guessing with that. Look, with the drop in carbon and nitrogen, once again, the nitrogen is an organic, in an organic form. And so as the carbon is mineralized, whether it's by heat or sunlight or whether it happens to be, the nitrogen is volatilized as well. And so that's something also that people need to be mindful of. You can have a load of a particular, let's say for example, a chicken litter delivered to your property, and if you don't apply it for a number of weeks, it could easily be significantly different than when it first arrived at your property, because it can dry out and things can happen. And that's one of the differences between using inorganic fertilizers and recycled organics. Inorganic fertilizers, you've got to remember, there's been massive amounts of research and money and experience gone into formulating the fertilizers, working out how they respond, testing that actual response all over the place. And we're really only just scratching the surface with the different ROs that we use. But part of the variability is how old is that material? How long has it been sitting there? How far along the composting process did it go? What were the animals fed as they were eating it? There were big differences, for example, between dairy manure and beef cattle manure. One from both of them were feedlot production systems. So, yep. Anyway,
I think that's a really good point, Mark, because I think sometimes people they know but they forget that inorganic fertilizers are somewhat stable product that has to conform to a particular set of parameters that are laid down. Whereas recycled organics, they have a lot of organic material and bugs and things in them, so they are constantly changing, and there's waxing and waning of things. So I think that's really important to remember.
Well, we see consistently 1, 2, and 3 orders of magnitude difference in, say, some of the contaminant levels in a couple of the RO materials over a year, and there's no reason to suggest that the carbon and the nitrogen will be non-variable as well. So if you're dealing with that as well as how it's treated before you get it and after you get it, you end up with a very complex system that you then have to base a farming or a management decision on. And these all add up to reasons why, ROs are not as readily accepted all over the place as they could be. In saying so, I think there are certain industries that just can't get enough of it, and that there may not be enough for the material to go around.
Yeah, thanks. Okay. Steve Kimber has another question here, which is one that had occurred to me as you were talking. He's called it application shock versus the trickle feed of amendments. So, given the issues that you had identified around risks, does a trickle feed not give the results that you've seen because of the smaller amount?
Steve. Yes and no. I think the part of the shock is actually ploughing the soil up and exposing everything when you're adding anything. It doesn't matter really what it is. I know that when we had the National Biosolids Research Program, we had a treatment where we added smaller amounts of biosolids each year rather than a one-off application rate, and we really didn't see any difference in yield response when it was applied in smaller amounts. Having said so, I really can't see why that's the case. I would have thought that to reduce shock, it's almost like a priming effect. You'll be priming the soil to accept the next lot of material that you're putting on the ground, and I think there's definitely some work that should and could be done in that sphere.
Okay, great. Yes. Then there's a few. And there was another question, kind of like that, around lower application rates from Matthew, and he said, how would you view the use of lower rates of ROs, say 4 to 5 tonnes per annum, as a standard practice in association with the synthetics, which I think you kind of mentioned before, with the objective to slowly improve organic matter over, say, a decade?
Yeah, look, I think I think you just got to be mindful of that some of that carbon may not stay there for long enough. That's why I think if you're actually using RO to boost carbon, you need to put a lot on straight away. I have talked to a number of growers who have used, certainly one grower who's used and it's got responses from an RO material at very low rates, and it turns out that person was also supplementing that RO with some urea. But if you're looking to boost carbon, I think research would show that, for example, a well-managed pasture system it was also a very good way of boosting soil carbon, and I think given that a lot of it will disappear when you put it on, you probably need, if carbon's what you're trying to do, you need to put a lot on. But certainly, if you're supplementing with inorganic fertiliser, that will help to mineralise the nutrients in that RO, but it will also mineralise the carbon as well. So it's really, I don't know that a carbon and a nutrient response are particularly compatible. I think that they're almost mutually exclusive, but yeah, look, I'm not a carbon specialist by any means, so that's my opinion on how I would deal with that one.
Yeah, okay, thank you. There's been a few questions about plastics as well. So someone's asked if, the microplastics question, and then someone's asked where the BPA comes from, so that's kind of linked in with that. And,
So what's the microplastics question?
Just if it's been looked at, have you found microplastics in recycled organics? Now appears that many organic composts processed at local council recycling sites will not be deemed organic because of the amount of microplastic. So that was a comment.
Yes, okay. Look, a lot of the organic contaminants we found in the recycled organic material we've looked at has directly come from plastic manufacture, and there was a lot of plastic in a couple of those materials that we found. I have not actually analysed the microplastics themselves, but I have analysed the soil in which the microplastics have been, and that's where the BPA has come from. There's also DHP, a whole range of other phthalates. Interesting, we found DEET as well, which is a component of insect repellent. But look, there have been a number of people that have done work looking at microplastics in soils. It's very difficult to do when you're actually adding an RO to then get the plastic out of the soil afterwards. So these tend to be model systems. And I think with a lot of the compounds that people look at, they're quite easy to look at in isolation under a sort of a model system. But once you start mixing them, it's quite difficult to clean up and actually do the analyses on those materials to actually get them out of the soil itself. Look, I do think that it is an issue with some of the RO material that we use, and one of the possible problems that could be is that there could be various antagonisms and synergisms with this whole range of chemicals that's coming out of the material that we're putting in, that one particular chemical may block the breakdown of another or vice versa. The other thing is that some of the breakdown products of these materials are actually more toxic than the original. But yes, there are definitely some issues with plastics in some materials, and once again, we possibly there could be issues with animal ingestion as well, but that's not something that we've tested. But certainly the organic chemicals that a lot of the organic chemicals that in some of the trials we've done recently have come from plastic manufacture and plastic breakdown.
Yeah, and would I be right in saying that one of the issues with microplastics, just as well as the physical contamination thing, is that what you've just highlighted, the breakdown products, the fact that they're smaller particles, they can interact with the environment and other things to produce more toxic elements. So that's the issue.
Oh, absolutely. And some of these are endocrine disruptors. Some of them are carcinogens. There's also some suggestion that microplastics can migrate from the animal's gut to the meat, and there's some work being done overseas about that, and it happens so it's of concern, and whether there's actually a lot of exposure for humans under the soil pathway, that's really not something I've looked at, but it's something to consider anyway.
Yeah, it's a new. So there's just a quick question about whether the manures that were used were grass-fed or grain-fed. Did you?
Okay, so one of the early tables I put on, I had the beef cattle and dairy cattle. They were both grain-fed feedlot materials. You tend to end up with slightly different phosphorus and nitrogen coming out of those animals, but they do tend to manage their inputs to a greater extent than if they were grazing, and that's about all I can tell you. We didn't have enough of replicates, for want of a better word, of feedlot based versus grain versus grass-fed. But I think that certainly the feedlot material, that they do regulate what goes into them a lot. They have much more control over what they eat.
And one would assume that, given that grass-fed animals are on paddocks and the manure is not collected, that most recycled organics would probably be from animals
from
that
feedlot.
Fed.
Yeah, absolutely. Yeah,
so,
absolutely.
Yeah. Okay, there's a few questions about the rate of IF and that work. I'm just trying to find. Virginia Brunton said, to what rate of IF are used as control, and did you do a regression analysis comparing nitrogen applied nitrification rate and yield responses to allow for the differences in N apply between the recycled organics?
Yes, we have done. We've certainly done contrast analyses for those treatments. The nitrogen was applied as per standard practice for those control fertiliser treatments, so roughly 100 to 120 units of N per year, including 2 urea top-ups, and that's just the standard practice for wheat.
Yeah.
If that answers that question.
I think so.
In terms of N mineralisation, that was pretty much a side issue. It's something that we could look at in more detail at some later stage, but it wasn't really the thrust of that particular work.
Yeah. And so following on from that, Michael said, do you know if the IF plots performed poorly compared to the control?
Yes and no. We did have a bad season to start with. The first season was quite wet. The next season, there was some frost damage. The IF was a little bit higher than, only just significantly higher than the control for that first season, and by the third season it was much higher than the unamended control. All the wheat yields, certainly for the second and third seasons for that grower trial, were much higher than the national average, and I guess that reflects how you manage an intensive trial as opposed to a paddock trial.
Okay, thank you. All right. Well, given that that's our hour that we usually go for, I will close this and just thank Mark very much for that presentation. Someone said it was very thought-provoking, and I think it is. It's not as simple as we would like to think, and yeah, we need to think about how we use these products to the best without some of those risks, or mitigating some of those risks. So thank you very much, Mark. Thank you everyone for attending, and we hope to see you next month. I can't recall who the speaker is at this point, but keep your eye out for your invitation, and we'll see you then. So thanks everyone.
Okay.
As many of you listening will probably know, the Soils Network of Knowledge webinars form part of a community of interest that DPI runs around soils, and we have some other products that help us with that as well. We have a Soils Unit Twitter account, and up there you can see our Twitter handle, and if you would like to tweet about today, please do. We would love to see you in the Twitter sphere, and there's a hashtag SNOC there that you might want to use. It makes it easier for us to find those tweets. And in addition to that, we also have All the Dirt, our quarterly newsletter, which has been running for over 10 years. And if you would like to subscribe to that, we also welcome anyone who's interested, and that's the subscribe address there underneath the banner.